So across the animal kingdom, there are quite a few different colors of blood. We have our animals, the majority of vertebrates, so that's pretty much, even fish and humans and fish, humans, apes, chickens have vertebrates. So they all use hemoglobin. So that's this chemical here that we've been talking about. This is that heme. This is the little heme molecule. This heme molecule is the one that's in each of those four quadrants of the protein. And so hemoglobin is a protein found in blood, built up from subunits containing hemes or hames. These contain iron, and their structure give blood its deep red color. Deoxygenated blood is a deeper red color, but it's not blue, remember. Meanwhile, you've got crustaceans, so spiders, crustaceans, mollusks, octopuses, and squids. They all use something called hemocyanin, and cyan is, of course, a type of blue, so that's probably where the name hemocyanin comes from. And they have copper instead of iron. So what do you know? It's pretty interesting. So unlike hemoglobin, which is bound to red blood cells, hemocyanin floats free in the blood. It contains copper instead of iron, and when deoxygenated, the blood is colorless. So it goes clear. But when oxygenated, it gives the blue coloration. So once again, it's a way of exploiting the fact that oxygen and metals like each other. It doesn't actually matter what kind of metal for the most part. So here we have iron, here we have copper. In some segmented worms and leeches, we end up with iron again, but in a different way. And so in this way, we get a sort of green color. So this is chemically similar to hemoglobin, you can actually see the structure is pretty similar. There's a little oxygen over here that I'm seeing. But otherwise, it's actually pretty much the same structure, isn't it, with one little oxygen on it. That can make a big difference, though. The blood of some species contains both hemoglobin and chlorocrucerin. Which is light green when deoxygenated. It is green when oxygenated, although when more concentrated, it appears a light red. And then we have violet in the marine worms, including the peanut worms, penis worms, and brachiopods. And so this is a very complicated molecule, much different, very, very different from those other ones. Well, it's actually not too different from this hemocyanin in terms of structure. Hemerythrin is only 1/4 as efficient at oxygen transport when compared to hemoglobin, and in the deoxygenated state, hemerythrin is colorless, but it imparts a violet-pink color when oxygenated. So there you go. Although we uniquely have red blood, we are kind of unique, and there are lots of ways to transport oxygen around your body. If anybody's a Star Trek fan, I think Vulcans have green blood. Yeah, I think it's Vulcans have green blood, and that's entirely possible. They might have some chlorine. So I thought it's interesting. I'll also post this one here. This is another one from that same website. They do the chemistry of blood. It gives you a little bit more detail about it. And here's hemoglobin is a protein built up of smaller subunits containing hames. They actually reused their old picture. Blood loss due to bleeding gradually turns brown as the hemoglobin is oxidized into methylhemoglobin. So that's why you'll see like a blood stain. If you ever have blood stain on clothes or whatever, it'll turn sort of a brown color over time. Just like rust turns brown, or rust is really brown in nature, you're oxidizing that iron and it's going to change color. There's a specific smell characterized with blood. This is caused by this compound. It's a very complicated name there. The metallic smell of metals and blood coming into contact with the skin is largely due to this subproduct that's produced from the reaction between them. And then today we're probably going to get to the blood typing section, and so we'll talk about that soon enough, so we can come back and look at this little diagram of the blood types. Does anybody have any questions about anything we've covered up till now? This is the beginning of class. This is a great time to talk about that. It looks like tomorrow's class we're going to spend some time on the digestion test and talking about that digestion test. So I will be giving you those marks back. I know everybody's been sitting on the edge of their seats. Like, I feel like that's what I should just keep pushing it back because my attendance is really good, and I'm like, I bet everybody's just waiting for that mark. So, but yeah, so tomorrow we'll go through the digestion test for sure. As I have my last person writing today, so that'll be great. All right, we have covered 3 of the 4 major components. So let's just quickly run them off. Red blood cells, what do they do? What's that? Yeah, hemoglobin, iron, and that's all for the surface area, so that you can carry oxygen gases from throughout the body, right? And why do we have to carry oxygen through the body? Because it's a good start. We can't live without oxygen, but specifically, what part of us can't live without oxygen? Cells. Ah, good. That's good. All right. So yeah. So red blood is there to bring oxygen around the body to feed the cells. Right. Cells need oxygen. Well, they don't need to eat oxygen, but they need oxygen to eat. So I don't know if is that a quibble? Maybe you need oxygen to eat. So without oxygen, a cell can't survive. So the red blood cells have to carry oxygen around the body because oxygen only enters your body through really one major area. Oxygen mostly comes in through your mouth, right? Yeah. There's a little bit of oxygen that comes in through your skin. It can be absorbed directly from the skin. So you can, there's some like famous examples of like things like, there's a really old movie, Goldfinger. Has anybody ever seen Goldfinger? It's a James Bond movie. No? Okay. So in Goldfinger, Goldfinger, the bad guy, he coats a woman in gold. And that's how she dies because her skin can't breathe. And they did do a Mythbuster on that, where they painted themselves with gold paint, and he actually got really uncomfortable. He sort of felt like he was suffocating after he was covered in gold paint. So I don't know. It's not conclusive about how much oxygen you actually absorb through your skin, whether you could suffocate someone by painting their skin, but certainly your body mostly absorbs oxygen through your mouth. Okay, what is the purpose of the plasma? Transport. Transport, what are some examples of things that they transport?
CO2.
CO2, yeah, great. Transport CO2. Oxygen, yeah, a little bit of oxygen, yeah. Bicarbonate, sure. Nutrients? Yeah, nutrients, amino acids,
and proteins.
Proteins, yeah.
Water.
Water. Yeah. Special proteins. Is that what it said? Or
yeah.
Special waste. Thank you. Sorry. Yeah. Special wastes, yeah, for sure. Nitrogenous waste, urea. Urea is an example. Great. So that's good. And then the last thing is the leukocytes. What do leukocytes do? Kill bacteria is a really good, important part. Yeah, so essentially kill bacteria, attack foreign invaders, those sorts of things. There's a whole bunch of options that we covered yesterday. Eating dead cells, that's another one. Okay, great. So then we have the last section, the last component of the blood that we're going to talk about, which is the platelets. And as is tradition, we have a better name, or a more formalized name, we have the thrombocytes. So platelets are a little bit funky. They are not really true cells. They're sort of chunks of cells. They have just some basic components. They don't have a nucleus. But they are built in the bone marrow. And they're these small cell fragments. They're essential to the first stages of blood clotting. So they come around. We talked about this in our positive feedback cycle. We did the thrombocyte a feedback cycle at one point. So their job is mostly to float around, and if they encounter some surface that's not smooth, they tend to explode. And when they explode, they release chemicals that will activate other thrombocytes, causing them to explode, and they'll also activate other chemicals in the blood that will cause some clotting to start to happen. So let's get into that. So they lack a nucleus. Their thin membrane is easily broken if exposed to any environment other than blood. This will release thromboplastinogen. Thromboplastinogen. Wow, what a great word. Okay, so we've got thrombo, right? So thrombo because it's from a thrombocyte. From a thrombocyte. So that's what a thrombo is. And then what is this word? What is plastin? How does that, like, what do you think about when you think about plastics? Yeah, and that's kind of the idea, is that it's a chemical that's going to cause a sort of plastic to form. So plastics are polymers. They're chemicals that sort of lay over each other, and that gives it that sort of flexible appearance. So they're going to release this thromboplastinogen, and that's going to be a factor that allows sort of the equivalent of a body's plastic to form. Or it's going to call over a bunch of fibers, and those fibers are going to bind themselves together, just like in a plastic. In a plastic, we add a emulsifying agent that connects the plastics together. So that's sort of the idea. It is in the thrombocyte, so it's a thrombo, and then it is forming, in a way similar to plastics, it's forming a clot. So blood clotting. So when a blood vessel is damaged by injury, it is no longer smooth, and this will break some of the platelets. So if we think about it being a smooth tube, if you suddenly have these sharp edges where you've got a tear, then you're going to have some unsmooth area there. So that breaks some of the platelets. Platelets breaking, of course, release the thromboplastinogen. That's what we just said, right? So we can kind of go like. And we'll do a cycle for this in another slide, I think. Actually, well, you have a picture of it right away. Okay. This thromboplastinogen gets transferred into thromboplastin, which is an enzyme that will convert prothrombin into thrombin using calcium. This thrombin will then convert fibrogen. Fibrogen is, of course, fibrous into fibrin. Fibrin is, whereas fibrogen, you can dissolve it in your blood's plasma, fibrin is considered insoluble. So insoluble means it's not dissolved in the plasma. So before you wouldn't have been able to see it, all of a sudden it'll sort of precipitate out of the reaction, out of the plasma. Fibrin is made of long fibrous proteins that form a network across the wound, and that slows the escape of blood from the wound and eventually stops it. So it's a bit of a process. So let's look at it here. So here is the damaged blood vessel. The injury of the vessel lining triggers the release of clotting factors. Clotting factors are being released because the platelets are exploding. And so that is our thromboplastinogen. So that's the word we're missing here. Thromboplastinogen. So that thromboplastinogen will combine with something that was already floating around in the plasma, which is the prothrombin. So this is just in the plasma. Same thing with this. This would be in the plasma. Okay. That's not exactly what I wanted, right? Okay. So that thromboplastinogen, it's going to activate the prothrombin, turning it into thrombin. Thrombin you're essentially activating an enzyme, right? And as we talked about, each enzyme has a very specific task. In this case, thrombin, the enzyme, once activated, is able to convert this other stuff, this protein stuff, fibrogen, into fibrin. Does something to the fibrogen that takes it from being in the solution to precipitating out of the solution. So one way to think about this is a precipitation reaction. This is not uncommon thing to do in chemistry. So if you're already in chemistry, you might have seen something very similar to this. So I'm going to make two little very quick solutions here. So we can think about it like this: that you have plasma, and it looks, in the case of the plasma, it should be clear. Sorry about that; that was a little white. But so we would have we have this clear plasma, and then all of a sudden, when the reaction happens, you have precipitation. So by this chemical reaction happening, all of a sudden, this yellow stuff is no longer part of the solution. Right? So if in the case of the fibrogen was clear, and then all of a sudden, because you created this other chemical and you've introduced it into the system, that fibrogen becomes fibrin, and now it's no longer part of the system. And if we wait till the end of the day, it should probably all drop to the bottom. You'll have a little bit of powder at the bottom. And so that's very similar to what's happening here. Fibrogen was happy enough to not be doing anything, just floating around in the solution. But once it's reacted with thrombin, you create fibrin. Fibrin, much like this lead iodide, is not going to be happy in solution. And it'll drop out. Where is it going to go? Well, it goes to the sides of the blood vessel. Down the middle of the blood vessel, there's blood pumping, so that pushes it to the side. It's no longer part of the solution. It's not getting pushed along with everything, and it sticks to the side. And you do that enough, it'll stick right where all that blood is flowing out and start to clog up that hole. Okay. So it's a precipitation of fibrin from solution. Any excuse to pull out chemicals. Good enough for me. Okay. So that fibrin gets all convoluted with the red blood cells that are trying to flow out this hole, plus all of the platelets that are trying to come out, and that forms a big clot. Now, of course, if you hear the word clot, you might recognize that blood clots aren't always found in places of damage. There are such blood clots that can form in a blood vessel, and they can be found in the heart or in the and cause a heart attack, or in the brain cause a stroke. Those are also formed in the same way of a collection of blood and vessels, or the thromboplastin has caused a conversion to fibrin somewhere in the blood, not necessarily somewhere you want it. Here's another version of this that I was supposed to skip over. So the same idea. Here you have a damaged platelet. It releases some thromboplastinogen. Here's your prothrombin that's just floating around. There's a calcium ion that's exchanged that turns it into thrombin. Thrombin is able to activate the fibrogen, turning it into the long fibers that can kind of cover up this hole in the cell. And when your blood doesn't clot, this is a disorder known as hemophilia. It's a disorder that is passed along through your genetics. Famously found in the royal family, like a really old royal family, like the Queen Elizabeth I or whatever. And her family, they had hemophilia, and there's a very well defined family tree that shows how hemophilia spread throughout that family. And hemophilia is a condition where somewhere in here there's a mistake, right? So a genetic mistake. So maybe the body can't produce platelets. It can't produce enough platelets, low platelet count. They can't produce prothrombin. That's just a protein of sorts. They can't produce, if the prothrombin is not formed, then the thrombin can't form. Or maybe there's difficulty in managing calcium, or difficulty in forming fibrogen. Typically I believe its platelet count is low in hemophilia. I think that's the most common kind, is your body's not producing enough platelets. So this is what it might look like if you zoomed in. This is the red blood cells trapped within what chemical is going to be this long fibers? Fibrin, yeah, very good. Yeah, so these long fibers, over here, maybe these little clots, these might be like platelets that are trapped. So this little thing here, that's probably a platelet that's been broken open. And then these are the thrombin, these are the fibrin that's been allowed to sort of fall in place. Okay, I have two. So on the Google Classroom, you should be able to find some links to my channel or to my playlists. We have a digestion playlist. In the digestion playlist, you'll find all the videos that we watched through the digestion. Apparently one has been deleted. That was mine. But also you'll see one video that I made during that digestion period. That was the last day of class, or like the Wednesday of the last week of class. And you can hear me. It talks. And then the other playlist that I have you linked to, if you click around, is the circulation playlist. And this is where I'm going to start putting stuff. So I put this is Monday's class here, April 1st. Here's April 2nd. This is the one that has no sound, unfortunately. I must have had some setting wrong, but you can see all the slides there. So if you don't like looking at the slides on the Google Classroom, you can use that link. I believe these are public, so you actually can find them. If you did a search for John Wren, you might be able to find them. Let's see if we can do that. I'm using incognito, so it doesn't have, let's see, John Wren. No, let's try biology. Circulation. Yeah, they're not.
I think you have to activate it on the settings.
Activate searching on my video, you think? Nope, doesn't fit. I don't exist yet. Okay, so not searchable, but the link is on the Google Classroom, so that'll be the best way to get there. I will also try to find some time today to put a link on my website. So there, if you are not able to get to the Google Classroom, you just don't like it, then on WEC's website, you can go to, this is a website, so the blogs Learn Everywhere. It's a big thing we're pushing right now. And so you should be able to go to science, and you can see my Bio 30S over here. And so right now it just says, go to my classroom, what are you doing here? But I do have some things on here. I have like a course outline, and that's about it. And then digestion, I did put up the PowerPoints for digestion, so if you wanted to see those. So, and now I'll add the link to the YouTube videos here as well. So I'll put those up there on the blog website, which you can access from the Learn Anywhere, which is an initiative we're trying to do, which is, but once again, it's going to encourage you to go to the Google Classroom because it has way more stuff and way more things you can do. All right, this was all to say that I'm just trying to get over to this.
In a first step towards clot formation, platelets are recruited to the site of vessel injury by now exposed molecules of the vessel wall, such as collagen and von Willebrand factor. This factor mediates the linking of platelets to collagen via a specific receptor in the platelet membrane. The resulting change of shape of the platelet from its resting state into the dendritic form indicates activation. The activated platelet in turn releases prothrombotic molecules such as adenosine diphosphate, ADP. By binding to its receptors, ADP induces aggregation. And recruits further platelets to the site. Thromboxane is another important mediator of platelet activation and aggregation. Under its influence, the platelets cross-link with each other. These interlocking mechanisms cause platelet activation to snowball. The clot grows rapidly. Activated platelets also trigger the coagulation cascade and thus the formation of thrombin. Thrombin, in turn, stimulates platelet activation even further, a continuous feedback loop. Additionally, thrombin induces the formation of fibrin for the mesh stabilizing the clot. The self-reinforcing process of platelet activation, crucial in the formation of blood clots, is an obvious therapeutic target in conditions caused by inappropriately triggered blood coagulation.
Okay, so once again, some of the problem with biology is that it goes deep, right? Like, so there we had a whole bunch of other stuff that they're talking about that we didn't really consider in our notes, and you know, I don't really want to add more layers to this. This is only one aspect of biology that we want to cover. So let's stick with the description we have, and so we're going to go with this cycle. This is the cycle we're going to be using, so watch out. If you start exploring the internet, you're going to find that there's even more parts to the development of clots, but this is certainly one of them. Now, I believe in the booklet I have a question there that I was initially going to include a slide on, but I think I skipped that. So I'm just going to pull that up right now. Right, so in the question 13 in your booklet, it says blood arteries are typically very smooth on the inside, but when someone has high cholesterol, it creates a narrowing of the arteries. This often increases the chance of blood clot forming, causing heart attacks or strokes. Why would narrowing of an artery increase the chance of blood clot formation?